So delighted to have a guest speaker today, Simon Buczacki, who many of you will know. He is a colorectal surgeon, quite a successful academic at Cambridge. I've come across him through Cancer Research UK. His very good work, which made him win 2 awards from Cancer Research UK, one initial clinician scientist, and then the second more recent and advanced clinician scientist. I won't steal his thunder, but he's got a very exciting research program, clinical practice focused on laparoscopic colorectal procedures. And he was telling me yesterday that he also leads something which is new and important from the US called Aerodigestive Cancer Program. Air and bowel, there we are. So maybe he'll tell us more about that. His lab program is mainly about the role subclonal interactions play on colorectal cancer cell identity and behavior. And we'll hear more about it, so I won't dwell on that. But a very warm welcome, Simon, and please, the floor is yours.
Thank you very much, Prof. Thank you very much for the very kind invitation to come and speak here. I'm talking on NETs, which really are not the main focus of my clinical practice or my scientific focus, but I thought they were interesting because they're interesting from a surgical perspective, and there's a lot of very interesting science around NET biology, which has been very poorly understood and poorly explored. So I thought I'd initially just give a little bit of a background to my clinical practice and the scientific practice and the setup we have in Cambridge, just to put everything into somewhat of a perspective. So our unit is similarly sized to the unit here in Oxford. We have 6 colorectal consultants who are doing purely colorectal surgery. We also have 3 emergency general surgeons who are also fully embedded into our department. So a quarter of their practice is EGS work and 3/4 of it is colorectal surgery, including resectional work. So we fully embed them. And then we have 1 full-time academic, myself. The department has a full spectrum of subspecialty practice. There's a pelvic exenterative service, which many of you in the field will know is led by Nikki Fernhead, supported by James Wheeler, who are both Oxford trainees originally, as well as John Morton and Justin Davis. Our pelvic floor service is run by John Morton and Michael Power. Early rectal cancer with TEMS is run by James Wheeler and Nigel Hall. We have an anal cancer service, a newly developed endometriosis service together with our gynecology colleagues, and also the NET service. And the NET service is really led solely by myself at the moment. We have a new colleague who will be starting next month, who will be joining me with NET resections, which will help because there's just not enough capacity for myself to do all of the NET resections as well as standard cancer resectional work. So similarly focused, I guess, to what you have here. We're based up on the Cambridge Biomedical Campus, and I don't know whether many of you have been up to Addenbrooke recently, but it is expanding at an ever-increasing rate. The Biomedical Campus is now the largest health-related biomedical campus in Europe and has a very strong cancer theme to it. Some of the new developments, so the hospital is based over here and sort of extends off the right-hand side of this photo. There's a lot of new scientific developments and industry on site. So this building in the shape of a chromosome is the new Laboratory of Molecular Biology, which opened about 4 or 5 years ago. This is AstraZeneca's worldwide headquarters for research and development, and this is Royal Papworth, which finally, after what seems like an eternity, has finally moved from their village site in Papworth to being on campus with us. This is the CRUK Cambridge Institute, which is where most of my research has been done to date, and I'm in the process of moving into this building, which is the Stem Cell Institute, which is being relocated from being in town up here on site. There's also the MRC Hutchison Cancer Cell Unit and the Cambridge Institute for Medical Research. There's going to be a new children's hospital developed on this site over this side, which has just got government support. And we're in the process of applying on this site to develop a cancer research hospital, but that will not have any surgery present within it. So there's clearly a very strong co-location of clinical and scientific disciplines on campus, which very much helps interdisciplinary approaches to research. We have strong funding from the BRC and Cancer Research UK, and we're a CR UK major centre. And as I say, with AZ being on site and MedImmune as well as ABCAM, we're having increasing amounts of collaborations with industry. The cancer focus is interesting and worth mentioning in that there's not a strong GI focus on site, and this is historical based on the PIs who are present there, and it's predominantly breast, ovarian and early detection. So my science, so I said my focus is mainly on colorectal cancer biology, and it's about understanding the interlinked behaviour between cellular identity and behaviour in cancer, and understanding why cells transition from one state to another. And we're particularly interested in cells that have received little focus to date, differentiated cancer cells, but we've identified that subpopulations of these can acquire proliferative capacity. We're also interested in tumour evolution. But today I'm going to be talking about small intestinal net biology, which I became interested in when I started to do net resections clinically. The techniques we use in the lab are very much based on using primary tissue, and that's why it's key to have your science based on a close location to where you're carrying out your clinical practice. We do a lot of organoid biology, using CRISPR targeting on normal organoids, single-cell work, so single-cell RNA-seq. We use a technique called lineage tracing, which I'll come on to shortly, and I'm increasingly interested in using game theory to understand clonal interactions. So, nets. To give you some background to them, they are very rare. They used to be called carcinoids, however, about sort of 10, 15 years ago, the nomenclature changed to neuroendocrine tumours. And this sort of figure on the left shows the common sites below the diaphragm to find nets. The three common places really are lung, small intestine, and pancreas. And today I'm going to be talking about the small intestinal nets. The reason I had a picture of Steve Jobs up is many of you will know that he died from pancreatic cancer, but he seemed to be in the news about his pancreatic cancer for many, many years. And if he had truly had a sort of pancreatic adenocarcinoma, he would have passed away much more rapidly. But he actually had a pancreatic net, and that's part of the reason that he lived for so long. And I gather he didn't even have conventional therapy for that. So, as I said, they're rare, but they're also very, very slow growing. So this table shows the 5-year survival rates for nets with both local and distant disease. And today I'm going to be talking about our small intestinal nets, and you can see that even if you've got liver metastases, with just standard of care treatment, there's a 54% 5-year survival rate. And this impacts very extensively your clinical decision making, and you can see that these vastly outweigh the equivalent survival rates for patients who've got adenocarcinoma with distant disease. So what do they look like? So this is a picture from a publication showing a typical net in the small intestine. I'd say that actually, in my experience, a lot of the primaries are much, much smaller than this, and you can't often even be able to detect them from the outside of the small intestine. And the only way you can actually identify them is through bimanual palpation. And they are often multifocal, so you'll have metachronous tumours throughout the ileum that you can detect by bimanual palpation. You might think, okay, well, that's pretty easy to take out, and the primaries are not the problem surgically. The problem is the mesenteric spread, and this is a resection specimen, not mine, of a patient who's had the primary removed, but also with the mesenteric lymph node spread. And you can see here that all of these small bowel loops have been pulled into this and cicatrized into this large mesenteric deposit, which has significant desmoplasia, making the resection of it challenging. It's also not uncommon to come across this upon opening up the abdomen, and you find bowel that you would never leave in if you were closing, and you'd think the patient was going to wouldn't survive, but they have chronic ischemia and you open up and you see that the bowel looks unbelievably unhealthy, and this chronic ischemia causes significant bowel wall thickening and subsequent obstructive-like symptoms. These tumours are really rare, so they if you take in context about colorectal cancer is going to affect between 1 in 14, 1 in 15 of us. These occur in about 1 in 100,000 patients. And because of this very slow and indolent growth, they often present very late with metastatic spread or mesenteric deposits. We probably all remember carcinoid syndrome from medical school, and they can also present with those symptoms. The treatment of them is complex and requires a very well-functioning MDT. Clearly, surgical resection is the mainstay of treatment with curative intent, but also involved is somatostatin analogues such as octreotide and lanreotide. And for liver disease, radiofrequency ablation, embolization, liver resection, or even indeed liver transplantation. Chemotherapy is only really of benefit when these tumours de-differentiate and start to become rapidly proliferative because of the nature of chemo only really working on cells that are rapidly dividing. So the science, though, is absolutely fascinating. So unlike colorectal cancer or other adenocarcinomas, we know so little about these tumours. We don't know what the cell of origin is, we don't know what the driving mutation is, we don't know what the aspect of epigenetics is on their development, or how these tumours evolve and progress. And there's lots of very low-hanging fruit related to the science of these tumours that make them very attractive to study. A little bit of more clinical detail briefly on how these tumours present. So they said they present late. They have obstructive symptoms rather than problems secondary to the primary, and the obstructive symptoms are secondary to ischemia. I would say that liver capsule pain isn't a common way that these patients present, but I have had 1 or 2 patients who've presented with liver capsule pain and deranged liver function tests, which have then subsequently been investigated and found to have metastatic disease. And this demonstrates the characteristic flushing associated with carcinoid syndrome. We use eNET guidelines to manage, work up, and develop subsequent follow-up pathways for all our patients. And if you're interested in NET, I very much strongly recommend looking at these guidelines, which are available online at the link below. They need to have a baseline chromogranin A, which is one of the best serum tumour markers to identify for patients who might develop further recurrence. A 24-hour urinary 5-hydroxyindole acetic acid is a prerequisite to identify and make the diagnosis. Staging radiologically is key, but most importantly in relation to identifying the anatomy of the vasculature that supplies the primary as well as the mesenteric disease. Octreotide scan is very important to identify occult peritoneal disease or liver metastases, but can on occasion be equivocal, and in those situations a PET scan can sometimes give you the key. In relation to surgical workup, an echo is also important to identify occult carcinoid valve disease, which again is not uncommon in these patients. And as I alluded to earlier, it's very important to have a functioning MDT with all of the associated subspecialty support. Staging is really quite simple. There are different staging types that have been put about. These are the eNET stagings. And as you can see, a tumour stage is based purely on size and invasion, and the N and M are binary 0 or 1, and you have your stagings of 0 to 4, so very straightforward staging. It doesn't really help, though, in the management, to be perfectly honest. So the general principles, as with any sort of solid organ malignancy, if they haven't got metastatic spread and they have limited nodal spread, the aim for curative resection is radical resection with curative intent. So resection of the primary and a radical lymphadenectomy to remove those mesenteric lymph nodes that are involved. There is some discussion as to whether this is safe to be done laparoscopically or open. The current recommendations, and what I would agree with, is that for anything other than resecting somebody who's got limited primary and mesenteric disease and they have metastatic disease in the liver, then only in that situation is a laparoscopic approach appropriate. And for all other approaches, this should be done via an open resection. And that's partly so you can make sure you get out all of the mesenteric disease, but also due to the multifocality of the primaries. And various techniques have been looked at, imaging and endoscopically, to identify whether to pick up these other primaries. But it's been shown that only by manual palpation has the highest sensitivity to pick up other primaries, so you have to do it open. If you've got metastatic disease, things become really quite complex, and it's partly related to the tumour biology and how slow growing these are. And you have to tailor what treatment you're going to be offered to how long the patient's likely to live with those disease, and they can live for many, many years with relatively stable liver disease. So there's various options for the liver, as I mentioned, and there are various palliative options for the primary and the mesenteric disease if these patients have obstructive symptoms. Chemo, as I mentioned, is possible if patients have highly proliferative disease. So this patient on the right is actually a consultant colleague who I operated on about 3 or 4 years ago, and she had a palliative laparoscopic right, and you can see she's got very extensive liver disease. She's now had stable disease, I believe, for about 3 years, and if that continues, then she would potentially be a candidate for our first liver transplantation for metastatic NET. So this is a sort of, I guess, one of the key slides in relation to the surgical decision making. And these are the questions that go through my mind when I see a patient in clinic who I'm trying to work up for whether they can be resected with curative intent. So as with any patient who you first see, the key thing is, are they fit for surgery, and are they going to engage with the process? And I think patient engagement with the decision-making process is absolutely key. They've got to buy in to all of the disasters that are associated with radical mesenteric resection. They have to understand, albeit there's only a small chance of it, if you work up these patients correctly, that things can go wrong in the operating theatre. They could end up with short gut, they could end up with a high output jejunostomy, they could end up on lifelong PN. And I don't think that you should embark on these operations where you're high up on the SMA and the SNV if the patient hasn't bought into that whole process. So the other question is how symptomatic they are from their primary and mesenteric disease. And then the question is whether or not this is resectable. Some studies have shown that if there is invasion of the mesenteric disease into the retroperitoneal structures, such as the duodenum, then that's a contraindication to surgery. I wouldn't say that's necessarily the case. I think there are patients, there's one I'll talk about next, where you can go ahead perform limited duodenectomy associated with the resection. You can't pick up the desmoplasia very easily on imaging, so that needs to be sort of taken into account. But the key bit here is in relation to the number of proximal branches of the SMA that are spared. So this is the Uppsala staging system, which I lifted from this chap's PhD thesis. It's a nice drawing he's got. And they describe these 4 stages of mesenteric disease, 1 to 4. Again, I think this somewhat oversimplifies the process, and I think you have to have a tailored, personalised approach to each patient. Generally, as a rule of thumb, if you have 3 spared jejunal branches, that should leave you with 100 centimetres of small bowel left. But apparently these are the emoticons for grimacing, which I didn't know. But it can be. It's not always an exact science. So ideally, you probably want more of those. But anything less than 3, then I think you're on a hiding to nothing with relation to not having enough bowel related. Debulking, which is something we shy away from for our bowel adenocarcinomas, can be used for patients who've got large disease, so very much applying the principles of gynae-oncology to an intestinal tumour. So I'll give you 2 examples of patients who we've operated on, just to sort of give you a flavour of some of the difficulties in relation to clinical decision-making in relation to patients and the surgical approaches that we use. So this first patient was a 70-year-old gentleman who presented initially with urological symptoms and then went on to have imaging for his hematuria. And he had a primary, which I've highlighted. Well, this is the mesenteric deposit in red. The primary was in his ileum. And you can see the bowel loops are tethered in. There's some fat stranding around the mesenteric deposit. And then green is his duodenum, which is sort of tented up, potentially involved in the mesenteric deposit. He was fully worked up according to ENET's guidelines. He had his urinary levels measured, and he was started on lanreotide. But he had no metastatic disease, and given that he was getting obstructive symptoms, we looked at him with a view to radical resection. So I mentioned before that the key aspect with these patients is identifying the arterial and venous anatomy associated with the deposit. So this reconstruction of the SMA demonstrates you can see that the vessels here at the distal part of the SMA are all being pulled in to the tumour, and the tumour is sort of just highlighted by this little area of calcification. The tumours don't appear very clear on CT. This is the same imaging from the same patient cross-sectionally. And on the right, what I've drawn on here is highlighted actually the area that was involved by the mesenteric and primary deposit, which basically were just one and the same. And you can see here we've got 1, 2, 3, possibly 4, but that's probably going to go in the resection margin. So three jejunal branches and a duodenum that was involved. But we deemed him suitable after getting a second opinion from Southampton for radical resection. So what's the surgical approach for these patients? Towards the latter end of my training, I was fortunate enough to go to visit this gentleman in Erlangen in Bavaria, which is a really beautiful part of Germany, I must say. So it's a lovely place to go to. It's a lovely university city outside of Nuremberg. And this chap here, who's unusually tall, and not that I'm small, he's unusually tall, is a chap called Werner Hochenberger, and he's the head of the department. And he developed this technique called complete mesocolic excision, which is and he used it for adenocarcinomas of the large bowel. And it's a much more radical lymphadenectomy and a very close, sharp dissection along embryological planes. And he's published his outcomes in various journals and improved 5-year survival rates for patients with even with early stage the old school BUCZ tumours to approaching almost 99%. It has come under some criticism more recently because of the morbidity associated with the procedure, but it was extremely useful to go out there and see him operate and start to learn the technique. So the technique that we use is applying CME, but for these net resections. And these two images from one of Hochenberger's descriptions of CME describe the essential steps. So here you've got the cava and the aorta, and this is the duodenum, not the colon. So you do a very radical full cockerization of the duodenum to bring it up out of the wound, so you can access on the right the root of the SMA and the SMV to identify whether you can actually resect this and leave enough bowel intact. So we mobilize everything, and then we obviously go above to then get access to this area here. So next case I was going to describe, this is again sort of shows some of the interesting intraoperative decision making that you have to make and the importance of working well with colleagues. So this was a 63-year-old lady who presented with typical features of carcinoid syndrome. She was investigated by our pneumonology colleagues and was found to have multiple liver lesions. Of note, her urine levels were extremely high, so that's a very high 5-HIIA level, and again a very high associated chromogranin level. She got obstructive symptoms, however, from her primary, and her actual primary mesenteric disease was relatively minimal, but you can see on this octreotide scan she's got significant liver disease. So because of her obstructive symptoms and her otherwise good health, we plan to do a palliative laparoscopic right hemicolectomy. So because of our high HIA levels, we gave them, almost all our patients have this, they have an octreotide infusion perioperatively to reduce the chances of a carcinoid crisis happening. We gave her a bolus followed by quite a high infusion, so this is 10 times higher than what people have previously described as the infusion required. And based on current literature, the incidence of carcinoid crisis with this degree of octreotide infusion is only about 2%. You can see what's going to happen. She got converted due to desmoplasia, and as soon as we started manually handling the tumour, she had a intraoperative PA arrest, and we felt that she'd had a carcinoid crisis. Looking under the drapes, everything, her skin was bright red. Our endocrine colleagues came into theatre and were in complete agreement that she'd had an intraoperative carcinoid crisis. So we were then left with this very awkward surgical dilemma of an otherwise fit lady who has had an arrest on the table. We'd mobilised the bowel by this stage, but hadn't actually divided any vessels or got the tumour out, with obstructive symptoms and really imminently about to obstruct. So what options are available? We could take her to ITU, bring her back, stabilise her, bring her back the next day. But the concern was, as soon as we start mobilising this tumour again, we're going to be in exactly the same situation. We could defunction her, bring a loop out upstream of the primary, which was one option. We could close her and not do anything and just stick her on SSAs and treat her palliatively. So lots of things to think about, and lots of discussion with colleagues, with our endocrine colleagues, with anaesthetists, with critical care support. And in the end, we did a very quick smash and grab right hemicolectomy and got the tumour out within sort of 5-10 minutes with relative stability, and obviously didn't join her up and gave her an endoileostomy. And she actually did very well following this, and she's gone on to have treatment for her liver disease. However, the moment this patient crosses the threshold into Addenbrooke, she seems to develop a carcinoid crisis. She even looks at like a medical instrument and she has a carcinoid crisis. So they tried to do an RFA on her, and even with the local anaesthetic, she started to develop carcinoid crisis-like symptoms. So she sees me back in the clinic and she's desperate to be joined up, but I'm less keen. So, in summary of our clinical experience, I'd say that it's really key to work as a team. You need a dedicated and experienced anaesthetist. You need a lot of endocrine support, octreotide infusions, and really I think open surgery is the key unless you're doing a palliative resection with minimal disease. We use CME resection, temporary slinging of proximal vessels to be absolutely certain that you're not going to lose a lot of small bowel. This is my friend, the LigaSure Impact. I think it's really useful with the desmoplasia in the mesentery. And you also need to have, aside from the LigaSure, you need to have good friends available for when things get hairy. So moving away from the clinical side, I'm now going to just touch on some of the net biology, which I said is really fascinating. And if there's anything you don't understand, do ask me. It's all really unknowns, as I alluded to earlier, and this is partly due to the paucity of tissue for analysis and how rare these tumours are. So most of the larger scale studies have only really been approaching around about 100 patients. The genomic studies, the very early ones, identified that there was somatic copy number aberrations and there was allelic loss of chromosome 18. But interestingly, when they looked at chromosome 18, where the usual suspects such as SMAD4 are located, none of these genes have been mutated. Probably the biggest study, which was this one published in Nature Genetics, which used whole exome sequencing on a panel of 100 small intestinal nets. In the keys in the title, they suggested that CDKN1B was a commonly lost truncal mutation in nets. But actually, when you get into the details of this paper, it's not common, and it was only actually present in around about 10 to 15% of their tumours. So I wouldn't say that it's a driver of nets. The role of epigenetics, be it methylation, histone modifications, microRNAs, is largely unexplored. There have been a couple of studies that have looked at methylation and identified this global hypomethylation associated with nets, but again, no specific genes. And microRNAs, like most microRNA papers, they come up with a list of 4 or 5 that they think are involved, but really that's about it. I've been interested in it in relation to the phenotype associated with nets, in that the characteristic secretory phenotype of the hormones and the relative quiescence of the cells that are present. And in particular, we've been interested in trying to understand what the cell of origin of nets are. And this paper from this American group was published in Gastro about 3 years ago, and gives some very interesting circumstantial data which agrees with what we think about what the cell of origin of nets are. So I'm just going to show you one of the figures from that paper. So this is some immunohistochemistry and in situ hybridization, which are 2 imaging techniques to identify what gene cells are expressing. And here's your tumor on the right, and they've got tryptophan hydroxylase staining in dark blue. and here they've just counterstained it in a red, or changed the colour to a red. So you can see that this is a crypt, which is present in the tumour, and they've got high tryptophan staining, as you'd expect in a NET. But also, in association with this, in the same cells, you've got very high expression of LGR5. And LGR5 is this ubiquitous stem cell marker. So if you're a stem cell biologist, the moment you hear LGR5, your ears prick up. The conclusion from this paper was that they felt that these familial nets arose from a subset of enterochromaffin cells, which are essentially enteroendocrine cells, that express reserve intestinal stem cell markers. So this is sort of very much in agreement with some of the data I'm going to show you next. So I'm going to show you a mixture of published and unpublished data. But before we go into the details of it, I'm just going to remind you of the cellular makeup present within the gut. So within the small intestine, we have crypts embedded into the wall of the bowel. We have villi projecting into the lumen of the bowel. Colon's exactly the same, but there are no villi. Within the bottom of these crypts is the stem cell zone, which we've known as the stem cell zone for donkey's years, 30, 40 years. And within the bottom of these, there are some undifferentiated cells, which are these slender green crypt-based columnar cells. There's also an undifferentiated, very slowly cycling cell termed the label-retaining cell in this characteristic +4 position from the bottom of the crypt. And then cells that are also present in the bottom of these secretory Paneth cells, and these are only really found in the small intestine. In the colon, you occasionally find them, but they're very essentially uncommon in the colon. And then you have absorptive enterocytes and lots of other secretory cells, so goblet cells, tough cells, and enteroendocrine cells. And these are the cells that are really very prominent within our nets. So what is a stem cell in the small intestine? And this is the seminal publication from Hans Clevers's group, who's based in Utrecht in Holland. And he used a technique called lineage tracing to very clearly identify that LGR5 was the bona fide marker of homeostatic stem cells in the gut. And lineage tracing is a technique which you do in mouse models, so animal models, and you develop this animal model whereby you give the mouse an injection of a drug, and that drug causes the activation of an enzyme. And that enzyme cuts and pastes in your DNA, or the DNA of the cell that it's activated in. And you can cause that enzyme to be activated only in specific cells because the enzyme's driven by a gene reporter. So let's say you're interested in saying, is LGR5 a stem cell marker? On the back of the LGR5 gene, you get activation of this enzyme called Cre recombinase. But only LGR5 expressing cells activate the enzyme. The enzyme then cuts and pastes and causes the expression of a reporter that you can actually visualize. Now, because that's a cut and paste, so it's a permanent genetic rearrangement, all of the daughter cells of that cell will inherit the activation of the reporter. So if a cell is a stem cell, then the daughter cells and the cells of the daughter cells and therefore and thereon will all activate, will all express the reporter. And if you then stain for it, you'll see clones of cells expressing the reporter, and that will show that they're a true stem cell. So that's the technique that Cleavers used, and he very clearly showed that indeed these LGR5 cells were stem cells. So the question was, what are these other cells? What are these the undifferentiated, slowly cycling cells? And various publications suggested various markers, BMI1, MTERT, HOPX, and LRIG1 as marking these undifferentiated, slowly cycling cells, which might be more likely to be the cell of origin of the nets. And Cleavers came back and then said, well, actually, that's all well and good, but all of your other markers actually overlap with LGR5, so in fact, you're just describing our LGR5 cells. So rather than using the technique that Cleavers used, we decided to approach this problem in a different way by isolating the slowly cycling cells based upon the nature of them being slowly cycling. So we developed this mouse model where you've got expression of yellow fluorescent protein tagged to a histone that's driven from an intestinal specific promoter, but as a pulse of expression. So here's a cross-section through the mouse gut. We've given the mouse the injection, and at T0, all of the cells of the intestinal epithelium express the yellow fluorescent protein. But it's just a pulse of expression, it's not on all the time. So with time, as the cells migrate off the villi and are lost or apoptosed and died, the signal becomes lost. But by 3 weeks, you can see at the bottom of the crypts, there are still these so-called label-retaining cells which retain the label and are therefore quiescent or dormant and haven't divided. And then you can use various techniques to pull these cells out and interrogate them for what genes they're expressing, whether they grow in culture and the like. And we, in collaboration with a group who are now based in Denmark, we could pull these out using a combination of two other markers, CD24, which marks the bottom of the crypt, and a lectin called Ulix lectin, which would rule out us pulling these long-lived Paneth cells. You might remember I mentioned earlier on. So the combination of these two markers allows us to pull out our label-retaining cells with very high affinity. So here are some actual images of the cells that you can pull out. Here are our label-retaining cells, which are CD24 positive, YFP positive, lectin negative. Here are the rapidly cycling cells, because they've in the bottom of the crypt, they're CD24 positive, YFP negative, lectin negative, and then the Paneth cells, which are double positive for them all. And as a result of using this mouse, we had sort of three different sort of publications describing the nature of the label-retaining cells and actually what was happening in the bottom of the crypts. And to summarize probably about sort of 8 years' work on one slide, we were able to show that indeed these LRCs expressed LGR5, but they also importantly expressed this other marker called PEG3 or PW1. They fell into a population called the side population. So the side population on this graph is marked by this black box, and you can see all the cells present in the grey colour fall into the box. And the side population is a technique that's been used in blood to identify stem cells in the blood, but is also clinically important because the side population is shown to overlap with the extrusion of DNA binding dyes, so thereby chemotherapy resistant. So they are potentially of interest from a clinical perspective. We found that during normal homeostasis, these cells weren't stem cells, and they were actually enteroendocrine and Paneth cell progenitors. And we did this by using short-term lineage tracing. I won't go through the details in this graph, but you have to just take my word for it. But interestingly, when we put these in culture, into organoid culture, which is when you can grow sort of mini organs in a dish in 3 dimensions, these cells were able to then grow into organoids, implying they had some degree of stem cell potency, still but latent. And then we finally resolved all of this by generating another mouse model whereby we could lineage trace, as cleavers did, but based for the first time in a mammalian system based upon how long a cell lived, and we're able to show that indeed when you injured the epithelium and the epithelium was regenerating, these cells changed fate from being a secretory progenitor to being a stem cell. So that was all very good. We've gone on to collaborate with a group in Barcelona led by Eduard Batil and identified 2 new markers of slowly cycling cells in the colon and the small intestine, whereby PTK7 and MEX3A, they can both generate multilineage clones, and they also both form organoids at high efficiency. But the interesting thing about our label-retaining cells is the relationship to enteroendocrine cells. And this red marker is chromogranin A, which is, remember, is the serum marker we use in our NET patients. And these label-retaining cells are destined to become enteroendocrine cells as well as the Paneth cells. And more recently, after our publication, there's been about sort of 6 or 7 other publications describing similar features of cells. So cells that are destined to become one type, but if you push them and injure the epithelium, they then acquire stem cell capacity. But of all these other populations, our LRCs are the only ones that are enteroendocrine cell progenitors. And combined with their very slow cycling dormant state, leads us to believe that they may well be the cell of origin of NETs. So are they? So this is unpublished data. Here are our LRCs. First of all, I mentioned that PW1 was a very specific marker of the label-retaining cells. And indeed, we've gone on to validate that in lots of ways and become very interested in PW1, which is also known as PEG3. It's an imprinted gene. The PW1 expression overlays very nicely with our LRCs. And looking at the whole gene transcriptomic data, so looking at what genes, all the genes that are expressed by our label-retaining cells, and then comparing it to previously published data sets. This is called gene set enrichment analysis. And the fact that this green line is all pushed over to the left-hand side, and you can see the bunching up of the black here. This is basically overlaying all our genes, which are the signature in our label-retaining cells, amongst a previously published data set of enteroendocrine cell progenitors, showing that indeed they have very, very similar gene expression profile. This is some single cell data whereby I'm identifying where cells that express PW1 lie in relation to all the cells that are present in the gut. These are the enterocytes, which I haven't highlighted, and then you can pull out using these single cell data. So these are individual single cells where these different populations lie. And you can just see up here, all of the red dots are present within the enteroendocrine cell pool, showing that the PW1 cells are indeed in the enteroendocrine cell population. But in fact, they're actually within the progenitor subpopulation of this group. So if you then take it to NETs, and then you say, well, are the genes that are expressed in our label-retaining cells overexpressed in NETs? You see the same thing. So gene set enrichment analysis demonstrates an enrichment of the signature within NETs. And turning that question on its head, looking at all of the genes that define the gene expression profile of NETs, again, these are all overexpressed significantly within our label-retaining cells. So we have a very strong suspicion that the label-retaining cells are indeed the cell of origin of the NETs. And the final sort of science slide is also, PW1 is of real interest because it doesn't just appear to be a marker of enteroendocrine cell progenitors. It seems to control their fate. So we've got a knockout mouse from a collaborator in Paris. These are the control slides. So this is PW1 expression. It is dark. You have to believe me, it's darker right at the bottom where the LRCs are. There is some low-level expression on the villi. And we can knock out the expression of it using tamoxifen to eliminate the gene. And doing transcriptomic data, so looking at what the genes change when you knock out the gene, what happens is you lose all of your MYC targets, which is basically saying that everything stops proliferating, and you lose the progression through the G2M checkpoint of the cell cycle. So again, things appear to slow down. This is validated using some staining for BRDU, which is basically a synthetic nucleotide which we can give to the mouse and is incorporated in cells that are actively dividing. Here's the quantification of that, where you can see that in the wild-type mouse, you've got higher levels of BRDU incorporation compared to the knockouts. But what is most fascinating is that when you lose expression of PW1, a large number of the cells change into enteroendocrine cells. You get much higher numbers of chromogranin-positive cells throughout the cryptVillus axis, and also tuft cells as well. So it's a very interesting gene. It's unfortunately very difficult to work with because it's expressed at very, very low levels. My collaborator in Paris has had 3 goes at trying to generate a mouse model where we can lineage trace from the PW1 cells, all unsuccessfully, but we're having another go at that ourselves, but it's of particular interest. So the summary of where we are with the NET biology, we're still very early stages. Clearly I've shown that the LRC work is very, it's all published and very clear, but the work with PW1 is still somewhat early in its stage. But I think it's a very interesting gene, largely unexplored in the gut. So what are the future directions for NET work in Cambridge in relation to the surgery and the science? We're in the process of applying for eNET Centre of Excellence status. As I said, I've got a colleague who's joining the department next month who will be, I'll be training up on NET surgery and will be joining me in helping out with getting through all our cases. I mentioned we're going to hopefully try and generate from the scientific side this PW1 CRE-RT2 mouse to enable lineage tracing from PW1 cells. We're generating a big biobank of organoids derived from the tumours and the normal matched normals. And we're also trying to model NET development by using CRISPR to target candidate genes from using the small intestine. So thanks very much, everybody, for listening. Obviously got to do my thanks. I have to thank Doug Winton, who's my scientific mentor in Cambridge and is an absolute fantastic world-leading stem cell biologist. My previous RAs, various core facilities in my old institute, colleagues, most importantly patients for their samples and consenting to our studies, Ruth Casey, who leads our NET team, Paul Rowe, who's my anaesthetist for the difficult NETs, Simon Harper, who's our HPB NET surgeon, who we often do combined cases with, and my various collaborators throughout the world. and funding bodies, Cancer Research UK and RCS, for funding my fellowship, and I receive core funding support from the Wellcome and the MRC at the Stem Cell Institute. Thank you very much for listening.